Clinical importance of alpha-1 antitrypsin and alpha-2 globulins. Basic information which is based on the last lecture, थोड़ा सा रिवीजन कर लें ताकि आपको पता चले कि क्या हम कर रहे किया पिछले उसमें और क्या अभी हम करने जा रहे हैं. So the basic information of alpha-1 antitrypsin based on the previous lecture. क्या होता है? एंटीप्रोटीनेज है. That is, प्रोटीन को टूटने से रोकता है, synthesized by the liver. It is a protease inhibitor. Just now we have seen that it inhibits the breakdown of the protein. And it inhibits the action of trypsin and elastase. Very important enzyme, elastase. Now from here we start this lecture. That was last slide was only a review जो कि मुझे इससे connect करना था आपको. That's why I have connected that slide with this. Clinical significance of alpha-1 antitrypsin. Increase of serum alpha-1 antitrypsin takes place in response to inflammation. Now what is the relation with alpha-1 antitrypsin and inflammation? It is a potent inhibitor of serine proteinase. That is the enzyme which removes serine from the protein chain. Okay? And thus have the ability to control the pro-inflammatory effects of serine proteinase. Serine proteinase क्या करता है कि ये सेरीन को सेपरेट कर देता है प्रोटीन चेन से. ठीक है? और इसका इन्फ्लेमेटरी इफेक्ट होता है और यू कैन से द प्रो इन्फ्लेमेटरी इफेक्ट होता है. अल्फा-1 एंटीट्रिप्सिन क्या करता है? इसको ख़त्म कर देता है. जब इसको इनहिबिट कर देता है, इट मीन्स दैट इट प्ले अ रोल इन स्टॉपिंग द और इनहिबिटिंग द इन्फ्लेमेशन. So it has the ability to control the pro-inflammatory effects, right? Once again, serine proteinase is an enzyme which removes serine from the protein chain and has the inflammatory effect. What is the role of alpha-1 antitrypsin? It inhibits serine proteinase. In this way, it controls the pro-inflammatory effect of the serine proteinases. Role in the emphysema of the lungs. Emphysema, I've told you that the alveoli are damaged and the oxygen capacity is removed. So how alpha-1 antitrypsin, it protects emphysema. Deficiency of alpha-1 antitrypsin has a role in certain cases. Approximately 5% of emphysema of lungs, it has a biochemical. What is the biochemical mechanism? Normally, alpha-1 antitrypsin protect the lung tissues from the injurious effects by binding with the proteases. Alpha-1 antitrypsin, it binds with the enzyme. Proteases are those enzymes which break down protein. So it binds with these enzymes. For example, active elastase. Active elastase have methionine at 358 position and it is involved in binding with the protease. Right? Protease, what is protease? That is the enzyme. यहाँ पे जो हमने protease कहा है, ये हमने active elastase को कहा है. Through the methionine which is present at 358 position, elastase, it binds with the alpha-1 antitrypsin. ठीक है? देखो active elastase का कौन सी चीज जो है वो alpha-1 antitrypsin से bind करती है? 358 methionine. That is the methionine which is present at the 358 position. And if this methionine is destroyed, then the action of the alpha-1 antitrypsin on the active elastase will be inhibited. So, active elastase through its 358 methionine, which is present at the 358 position. It binds with the alpha-1 antitrypsin, and in this way elastase it becomes inactive. ठीक है, ये जो कॉम्प्लेक्स बन गया, this complex is inactive and no proteolysis of the lungs takes place, therefore no tissue damage takes place. ठीक है, नो लंग्स की प्रोटियोलाइसिस होगी एंड नो टिश्यू डैमेज टेक्स प्लेस. When alpha-1 antitrypsin is deficient or absent, so the above formation of the complex does not take place and the breakdown of the lungs, proteolysis of the lungs takes place and the damage occurs. Relation with smoking. Just now I have told you that the methionine which is present at the 358 position is involved in the binding of the alpha-1 antitrypsin with the elastase. Now, smoking, it destroys this methionine. So, the complex of alpha-1 antitrypsin and the elastase is not formed, right? And the elastase is remain active and thus proteolysis of the lung take place and the tissue damage occurs, accelerating the development of the emphysema. Role in liver cirrhosis. Role of alpha-1 antitrypsin in liver cirrhosis. Juvenile hepatic cirrhosis, that is, will take place in the children, has also been correlated with alpha-1 antitrypsin deficiency. In this condition, molecule of toxic, insoluble antitrypsin Z, it aggregates and accumulate in the endoplasmic reticulum of in the cells of the endoplasmic reticulum of hepatocytes. And this leads to the inflammation. What happens in the juvenile hepatic cirrhosis? Toxic insoluble ATZ protein aggregate in the endoplasmic reticulum of the hepatocytes, and this leads to inflammation. The hepatocyte cannot secrete this type of mutant alpha-1 antitrypsin. This is the mutant form, antitrypsin Z. It is the mutant form and the hepatocyte cannot secrete this particular type of alpha-1 antitrypsin. Aggregation is due to the formation of polymers of mutant alpha-trypsin. Mutant alpha-trypsin कौन सा है? ATZ. The polymers, they form a strong interaction between the specific loop in one molecule and a prominent beta-pleated sheet in another. So one loop and the beta sheet of the other molecule, they form a complex, and this is known as a loop sheet polymerization. किस तरह से एग्रीगेशन होती है कि एक स्पेसिफिक, एक मॉलिक्यूल का एक स्पेसिफिक लूप और दूसरे मॉलिक्यूल की प्रोमिनेंट बीटा शीट, ये पॉलीमराइज हो जाते हैं, एग्रीगेट फॉर्म कर लेते हैं, एंड इन दिस वे द लूप शीट polymerization takes place. The aggregate leads to the damage to liver cells and leading to hepatitis and cirrhosis. Accumulation of massive amount of the collagen takes place, and the accumulation of the massive amount of the collagen, this leads to fibrosis. Role as a tumor marker. Alpha-1 antitrypsin has been used as a tumor marker. It is increased in the germ cells tumors of testes and ovary. Next function is that it also serves as an inhibitor of fibrinolysis. Alpha 1 antitrypsin is one of the most important inhibitors of fibrinolysis, along with alpha 2 antiplasmin and anti alpha 2 macroglobulin. So, all these inhibitors they block the action of plasmin on fibrinogen. After completing alpha 1 globulins, we start now the alpha 2 globulins, right? This is the second class of the globulins. Alpha 2 globulins, it is of two types, ceruloplasmin and haptoglobin. Ceruloplasmin, you have seen when we were studying the chapter of iron metabolism. Ceruloplasmin, it converts iron from its ferrous form to the ferric form. So it is a copper containing alpha 2 globulin. It is a glycoprotein. Glycoprotein means carbohydrate is combined with the protein. It has 8 sites. This slide is very important for the one incorrect answer. It has 8 sites for binding copper. Contain about 8 atoms of copper per molecule, and the copper is present in the cuprous form. Half of the copper it is present in the cuprous form, and half of the molecules they are present in the cupric form. It means 4 are present in the cuprous form and the 4 are present in the cupric form. It carries 0.35% copper by weight, and the normal plasma concentration is approximately 30 milligrams per 100 ml of plasma, and about 75 to 100 micrograms of copper may be present in the 100 ml of plasma. It has activities of copper oxidase, histaminase, and ferrous oxidase. Copper atoms are attached to a protein known as the apoceruloplasmin. So this is the protein, and apoceruloplasmin is a protein part, and when copper comes and attaches with this protein, then ceruloplasmin is formed. Level of ceruloplasmin, it varies with age and sex. There is low concentration at birth, gradually increases, and slowly continues to rise with the age afterwards. Adult females have higher concentration than the males. Functions of ceruloplasmin. Although ceruloplasmin is not involved in copper transport, it contains too much copper, but it is not involved in copper transport. 90% or more of the total serum copper is contained in the ceruloplasmin, but it does not transport copper. Its main function is as ferrooxidase and help in the oxidation of iron from the ferrous form to the ferric form, which then carried by transferrin to the tissue cells. Just revise your lecture on the iron transporters. It is increased in pregnancy, inflammatory processes, malignancy, oral estrogen therapy, and contraceptive pills. Contraceptive pills is for the birth control. Decrease is found in Wilson's disease. What is Wilson's disease? It is a genetic disorder in which excess of coppers build up in the body, and symptoms are related to the brain and liver. Menkes disease, it is also known as a Menkes syndrome, is caused by mutation in genes coding for copper transport protein, leading to copper deficiency. Genes are not present, copper transport protein is not present, and therefore copper deficiency occurs. And the characteristic findings include kinky hair, small hairs, growth failure, and nervous system deterioration. This is to discuss these diseases is not our field. It is included in the pathology, but since I have come across these terms, so in few lines, I have explained to you the disease or the syndrome.